Hello everyone. Today it's my pleasure to introduce our work. Common mycorrhizal networks enhance defense responses against pathogens in neighboring plants. Arbuscular mycorrhizal AM fungi are one of the most functionally important groups of soil microbes. AM fungi can enhance the tolerance of the host to pathogens. AM fungi lack specificity to their host plants. Thereby external mycelia known as common mycelial networks CMNs connect the roots of various plant species, as well as individuals of the same species. The CMNs can also transfer stress defense signals. This transfer induces defense responses from the neighboring plants and can effectively serve as an early warning before the stressor fully manifests. The previous studies focused on the transfer of defense signals mediated by CMNs between plants of the same species. However, mostly different species of plants coexist in natural ecosystems, and CMNs connect these diverse plant species. The phyllosphere microbiomes can provide a defense against plant pathogens. AM fungi can modulate the phyllosphere microbiome. However, existing studies have not yet explored the changes in the phyllosphere microbiome modulated by CMNs, particularly during pathogen invasion. Here, we aim to explore the effects of pathogen infection of the donor white clover on the defense compounds and phyllosphere microbiome of the receiver ryegrass linked to the CMNs, while revealing the correlation between defense compounds and the phyllosphere microbiome. Highlights: CMNs provide a channel for interplant communication, which enables plants of different species to prepare for pathogen infections without direct exposure. CMNs change the microbial abundance and community structure of the neighboring plants following pathogen infection in the donor plants, enhancing microbial interaction and network complexity in neighboring plants. This activation may trigger defense responses in the receiver plants against potential pathogens, which enhances the system's ability to resist infections. The results of mycorrhizal colonization showed that CMNs were successfully established between donor clover and receiver ryegrass. Elevated activities of CAT were observed in the donor clover infected with the pathogen and the receiver ryegrass, connected through CMNs. This result suggests that the CMNs may transfer profiles related to defense enzymes at the plant community levels, and possibly enhance the resistance of plants in ecosystems. Pathogen infection in donor clover changed the fungal diversity ACE index, genus and species richness and bacterial community structure of the receiver ryegrass phyllosphere. The number of shared fungal ASVs between the donor and ryegrass linked by the CMNs significantly increased after the donor clover was infected with the pathogen. The plant defense enzymes PPO and CAT were significantly correlated with the diversity of phyllosphere fungi. Significantly enriched biomarkers in ryegrass linked to diseased clover through CMNs include Vitiacozyma and glomeromycetes, and their relative abundance is significantly positively correlated with CAT activity, which may play a role in resistance to the pathogen. The donor clover infected with the pathogen exhibited a more complex fungal and bacterial network structure compared to the uninfected clover. Notably, a significantly more intricate fungal network structure was observed in ryegrass connected to the clover infected with the pathogen than in both the non-CMNs and ryegrass connected to healthy clover. Therefore, the microbial network complexity increased after the clover was infected with the pathogen, which subsequently affected the adjacent ryegrass through the CMNs. This ultimately enhanced the complexity of the ryegrass microbial network and improved the stability of the system. Summary: CMNs change the microbial abundance and community structure of the neighboring plants following pathogen infection in the donor plants, which further enhances microbial interaction and network complexity in neighboring plants. This activation may trigger defense responses in the receiver plants against potential pathogens, which enhances the system's ability to resist infections. Further studies should be conducted to explore whether these changes mediated by CMNs increase the resistance of the interconnected plant communities and whether these phenomena occur in natural settings.